dear students. Most of the reagents used in the scientific laboratory are in the form of solutions that need to be prepared or purchased. For some cases, the exact concentration of the solution is not important, while for other cases, the concentration and the method of preparation of the solution needs to be as accurate as possible. Today, we will learn how to make solutions in the laboratory. But before that, we need to get acquainted with the glassware used for this purpose. This is a glass pipette that is used to transfer fixed volumes of liquids from one container to another. The pipette that I am holding at the moment can measure up to 25 ml of the liquid. Remember that you should never use mouth pipetting for transferring liquids. Always use a pipette sucker, pipette filler, or pipette bulb for transferring the liquid. This is a volumetric flask that can be used in the preparation of fixed volumes of solutions. These flasks can only measure up to a single volume. For instance, this flask can be used to prepare a solution of 500 ml only. This single mark here indicates that when volume of the solution reaches up to this mark, the volume will be 500 ml. These flasks are available in 100 ml, 250 ml, 500 ml and 1000 ml capacities. This is a measuring cylinder. Measuring cylinders can also be used for preparation of solutions and come in various sizes with different capacities. For instance, this is a 10 ml measuring cylinder, this is a 100 ml measuring cylinder, and this is a 500 ml measuring cylinder. This is a conical flask, and like other glassware, this also comes in varying sizes of different capacities. This is a beaker and is also available in different capacities. Remember that beakers and flasks may be used while preparing solutions but are never used to define the total volume or the final volume of the solution. Whenever preparing solutions, always raise the final volume in a narrow-necked glassware, such as a volumetric flask or a measuring cylinder. Also note that when preparing solutions, always use the upper meniscus for colored liquids and lower meniscus for colorless liquids to avoid parallax error. Concentration is a measure of the relative proportion of solute and solvent present in the solution and can be measured in terms of percentage, molarity, molality, and normality. Now that we have learnt calculations, we will be preparing a 2% solution of sodium chloride. Whenever using reagents or salts in the laboratory, ensure that you read the expiry date on the bottle and read any warning or hazard instructions that are given on the label of the bottle. To make the solution, we will first weigh 2 grams of sodium chloride . transfer this to a clean beaker. Next, add approximately 30 ml of distilled water. Since we're making a total volume of 100 ml, we can use any volume less than 50 ml at this stage. Mix the solution to dissolve all the salt. Once the salt has dissolved completely, transfer this to a measuring cylinder. Next, raise the volume of this solution to 100 ml using distilled water. As told earlier, read the lower meniscus for colorless liquids. Once the solution is ready, transfer it to a labeled bottle and store it. Now that we have learned the calculation, we will prepare 50 ml of 0.5 molar sodium hydroxide solution. For this purpose, we will be using aluminium foil to weigh the sodium hydroxide. sodium hydroxide pellets get hydrated very quickly, so this step needs to be done quickly. Add approximately 20 ml of distilled water to this pellet. Mix the solution to dissolve the pellet. You will notice that heat is liberated as sodium hydroxide dissolves in the solution. If you are preparing large volumes of sodium hydroxide solution, you can do this step using a magnetic stirrer to avoid splashing. Once the pellet has dissolved completely, transfer this solution to a measuring cylinder and raise its volume to 50 ml using distilled water. Once the volume of the solution has been made up to 50 ml, transfer it to a labeled reagent bottle. Now, we will learn how to make dilute solutions from concentrated solutions. To prepare 0.1 molar sodium hydroxide solution from 0.5 molar sodium hydroxide solution, we will first take 10 ml of the stock solution, that is 0.5 molar sodium hydroxide solution. Now, raise the volume of this solution to 50 ml using distilled water to prepare 0.1 molar sodium hydroxide solution. Transfer this solution to a labeled reagent bottle. Now, we will learn how to measure the pH of any solution using a pH meter. Carefully remove the bulb of the pH meter from its solution. Immerse it in distilled water. Ensure that at any point during measurement, the bulb of the pH meter does not come in contact with the walls or base of the container. Then, wipe the bulb of the pH meter using a lint-free tissue paper. Pour the test solution in a beaker. And immerse the bulb in this solution. Allow the reading on the pH meter to stabilize. The pH of this solution is 12.68. Once you have measured the pH, Wash the glass bulb again in distilled water. The bulb of the pH meter is very sensitive and needs to be handled carefully. Once you have taken the reading, place the bulb in its solution again. This container contains a solution of potassium chloride. We will now learn the concept of normality. Normality is generally used for acids and bases. To calculate the normality of an acid, we will need information from its bottle. This is a bottle of sulfuric acid. Check the percentage purity of this sulfuric acid. The percentage purity mentioned on the bottle is 97%. Next, we need to check the specific gravity of this sulfuric acid. Specific gravity in simple terms is basically the density of this solution relative to water. A specific gravity greater than 1 indicates that this solution is heavier than water. The specific gravity mentioned on this bottle of sulfuric acid is 1.84. The portion about sulfuric acid has to come before the part about measuring pH.
Today, we will discuss about DNA extraction. Extraction and purification of DNA is required for various molecular biology techniques. DNA can be extracted through different methods, including organic method, inorganic method, and even through commercially available kits. We will extract the DNA by the organic method. Before proceeding to the experiment, we should know about the sources of the DNA, materials and the instruments required, and the basic steps that are involved in the DNA extraction. In the sources of the DNA can be extracted from human tissues, blood sample, plant tissue sample, bacterial cultures, spores, soil sample, and even forensic sample. In materials and instruments, we need different reagents like lysis buffer, proteinase K enzyme, sodium deutesyl sulfate, ethanol, and isopropanol. In instruments, we need pipettes, pipette tips, vortex, centrifuge machine, and deep freezers to store the DNA. Now, in the basic steps of DNA extraction, there are 6 core steps involved. The first step is the cell lysis. Second step is the digestion. Third is the phase separation. Fourth step is the DNA precipitation. Fifth step is the washing by the ethanol, and then air drying and the storage of the DNA. First of all, we will take the sample to extract the DNA, and the sample that we have is the human blood sample. We will take 200 microliter of we will take 200 microliter of human blood and we'll add in each tube. After the addition of 200 microliter blood sample, we will add 1000 microliter of lysis buffer. So we will pick the pipette which is 100 to 1000 microliter and will set on it 1000 microliter. Now we will add the 1000 microliter lysis buffer in each sample. After the addition of 1000 microliter lysis buffer, we will vortex the sample for 5 minutes. This is vortex machine, and we will now vortex the sample to homogenize the mixture. After the vortex, we will centrifuge the sample at 13,000 rpm for 15 minutes at 4 degrees Celsius. We will keep the samples in centrifuge machine. it should be remembered that while keeping the samples in centrifuge machine should be balanced from each side. Centrifugation will be done at 13,000 RPM for 15 minutes at 4 degrees Celsius. So we will set the centrifuge machine at 13,000 RPM at 4 degrees Celsius for 15 minutes and start. After the first washing, we will discard the supernatant, and at the end of the tube we have a pellet which contain our DNA. Pellet is actually a collection of cells where the DNA have, and after these steps we will go for the cell lysis through the digestion buffers. This was our first washing. We will discard the supernatant, preventing the pellet. Now we will again add the 1000 microliter of lysis buffer, and we will again do washing until the pellet become colorless. For the second washing, we will again add the 1000 microliter lysis buffer. After the addition of 1000 microliter lysis buffer, we will again move to centrifuge machine and will centrifuge the samples for 15 minutes at 13,000 rpm at 4 degrees Celsius. After the second washing, the results will be like this. You can see the palette has become somewhat colorless, but we will again do the third washing until the palette will become totally colorless. We will discard the supernatant, preventing the palette. Again, we will add 1000 microliter lysis buffer for third washing. Now again for third washing, we will centrifuge the sample on the same conditions as previous, like 13,000 rpm speed at 4 degrees Celsius for 15 minutes. After third washing, the results will be like this. The palette will become colorless. We will discard the supernatant, preventing the palette. After the 3 steps of washing, when the palette will become colorless, we will add 20 microliter of proteinase K. The basic function of the proteinase K enzyme is to digest the proteins. As we know, the DNA is associated with histone proteins for its packaging inside the nucleus of the cell. So the proteinase K will digest the proteins, which will lead to the specific extraction of DNA. We will take that pipette within a range of 20 microliter in order to add the proteinase K in the solution. After the addition of proteinase K, we will add the 80 microliter of sodium dodecyl sulfate, which is commonly known as SDS. The basic function of SDS is also to denature the proteins. Basically, SDS is a detergent that imparts negative charge to all proteins. And we know that DNA also contains the negative charge. So when all the proteins get the negative charge, they will get repelled from the DNA and will be separated from the DNA. So SDS is also used to digest the protein. We will take the pipette within a range of 10 to 100 microliter in order to add the sodium dodecyl sulfate. We will set the pipette at 80 microliter. After the addition of 80 microliter SDS, we will add 250 microliter of buffer A1, which will be used to stabilize the DNA at the required pH. We will take pipette of range 100 to 1000 microliter and will set on 250 microliter in order to add the buffer A1 in the solution. After the vortex, we will incubate the samples at 58 degrees Celsius for overnight digestion in water bath. The basic purpose of overnight digestion is to obtain the DNA purified from the proteins. So the proteins will get denatured and digested overnight with the help of SDS, proteinase K and the buffer A1. We will keep the sample for overnight digestion in the water bath at 58 degrees Celsius. Water level should be enough that our sample should be dissolved from the bottom. In the part B, first of all, we will pick the samples from water bath and then add the phenol chloroform isoamyl alcohol. Now, this is our sample after the overnight digestion. After the overnight digestion, we will complete the process of DNA extraction, which is the part B. Now we will add the phenol chloroform isoamyl alcohol in all the samples. The basic purpose of phenol chloroform isoamyl alcohol is for the phase separation. The basic purpose of isoamyl in the phenol in PCI is to prevent the foaming formation in the interface of the layers. Now we will add the 300 microliter of phenol chloroform isoamyl alcohol in all the samples. We will set the pipette at 300 microliter. Phenol chloroform isoamyl alcohol, known as PCI, is an organic solution which is light sensitive. Therefore, we wrapped it in a aluminium foil. Actually, the PCI contains the three components: phenol, chloroform and isoamyl alcohol. The ratio of each component is 25:24:1. 25 parts are of phenol, 24 parts are of chloroform and 1 part is of isoamyl alcohol. We will add the 300 microliter of phenol chloroform isoamyl alcohol in our sample. After the addition of phenol chloroform isoamyl alcohol, we will vortex the sample for 5 minutes. After the vortex, we will centrifuge the samples at 13,000 rpm at 4 degrees Celsius for 15 minutes. After the centrifugation, you will see that there will be the phase separation in the tube. Because the PCI was an organic solution, so it separates the DNA from the protein and make the distinct layers. The lower layer, which is at the bottom, contains the debris and the proteins. Debris are the worn out parts of the cell, and the upper aqueous phase contains our DNA. We will transfer the upper aqueous phase into the new Eppendorf tube. After the transfer of upper aqueous phase to the new tube, we will then add the isopropanol to precipitate the DNA. The basic function of the isopropanol is to precipitate the nucleic acid. So here in this case, when we are extracting the DNA, the isopropanol will precipitate the DNA. Now we will add the equal amount of isopropanol in the sample. So equal amount means the volume that was of the upper phase, we will add the same volume of the isopropanol. The volume of the upper aqueous phase was 500 microliter. So we will add the isopropanol 500 microliter in the sample. In order to add the isopropanol, we will take a pipette of range 100 to 1000 microliter and set at 500 microliter. After the addition of isopropanol, the DNA in the tube will get precipitated and we can analyze the threads of the DNA in the tube. After the addition of isopropanol, we will incubate the sample for 5 minutes at room temperature and then we'll proceed to the centrifugation with the same condition, 13,000 RPM speed for 15 minutes at 4 degrees Celsius. After the centrifugation, the DNA will be settled down and we will discard the supernatant and then wash the pellet with the chilled ethanol. Now we will add the 500 microliter of ice chilled ethanol in order to wash the DNA pellet to remove the remaining contamination. We will set the pipette at 500 microliter in order to add the ice chilled ethanol. After the addition of ice chilled ethanol, we will again centrifuge the sample at 8000 RPM for 7 minutes at 4 degrees Celsius. After the centrifugation, we will discard the supernatant and then air dry the pellet. After the air drying of samples, I have added the nuclease-free water and now we will store the DNA at -20 degrees Celsius. While working in the molecular biology laboratory, we extract different kinds of biological molecules. In order to analyze the expression of genes implicated in different disorders or the detection of RNA viruses like coronavirus or any other downstream technology, we first have to extract the RNA. There are different methods to extract the RNA, like the kit method, triazole method, or the manual method. But the triazole method is the most commonly used method while performing the extraction of RNA in molecular biology laboratory. So in this practical, we will demonstrate that how to extract the RNA from blood using triazole method. Let's perform the experiment. The major reagents and materials required for RNA extraction are blood, triazole reagent. Triazole reagent is a light sensitive chemical and it preserves the integrity of RNA. Next is chloroform, isopropanol, ethanol, and finally nuclease-free water. First of all, 250 microliter blood sample will be taken in Eppendorf tube. After the addition of blood sample, 750 microliter of triazole reagent will be added. Once you will add the triazole reagent, Eppendorf tube will be inverted to mix the samples homogeneously. Next, 200 microliter of chloroform will be taken and add in the Eppendorf tube. The function of chloroform is to separate the RNA from other cell contents and proteins. After the addition of chloroform, Vortex the opened off tube for 1 minute. In next step, we will centrifuge the sample at 13,000 rpm for 15 minutes at 4 degrees Celsius. After the completion of centrifugation, you will take out the opened off tube . As a result of centrifugation, you will observe 3 layers. Upper is the aqueous phase that contain the RNA. Second is the interphase, and third are the impurities. Now we will transfer the aqueous phase to a new opened off tube . It should be noted that the tip of the pipette should not touch the interphase. You will just pick the aqueous phase that contain the desired RNA molecule. After the transfer of aqueous phase to new opened off tube, we will add the equal volume of isopropanol in the opened off tube to precipitate the RNA. Similarly, after the addition of isopropanol, we will again centrifuge the sample at 13,000 rpm for 15 minutes. After centrifugation, you will observe the pellet in the bottom of the tube and you will discard the supernatant, preventing the pellet. Now we will wash the RNA pellet with ethanol to remove the impurities. So 5 to 600 microliter of 70% chilled ethanol will be added into the Eppendorf tube. After the addition of ethanol, we will again centrifuge the samples at 13,000 rpm for 15 minutes. After the centrifugation, discard the supernatant. Finally, we will add nuclease-free water into the Eppendorf tube after air drying the ethanol. The nuclease-free water will protect the RNA. Finally, after the addition of nuclease-free water, we will store the RNA at -80 degrees Celsius. After the completion of DNA extraction, we can confirm the presence of DNA through gel electrophoresis. So in this practical, we will demonstrate the examination of DNA through agarose gel electrophoresis. Basically, agarose gel electrophoresis is a process which is used to separate the DNA by size using the electric field that induce the negatively charged DNA molecules to migrate towards the positive pole through the matrix of agarose. Now, before proceeding to the experiment, we will look at the required materials and instruments. For the agarose gel electrophoresis, we need DNA template, (6x) loading dye, which is bromophenol blue, ethidium bromide, which is a fluorescent dye used to visualize the DNA under UV light. Agarose powder, (1x) TAE buffer, pipettes, measuring cylinder, conical flask, gel caster, gel tank, and electric supply.
First of all, we will weigh the agarose powder with the help of weighing balance. We will weigh 0.8% to 1% agarose gel. For this purpose, we will weigh 1 gram of agarose and will add into 100 ml of TAE buffer. After weighing, agarose powder will be added to the conical flask. Next, measure the 1x TAE buffer in the cylinder and add into the flask. Mix the flask contents well and kept in oven for 1 minute. After 1 minute, take out the flask with the help of heat-resistant glove and see whether contents have been dissolved or not. If contents are not dissolved, place the flask again in the oven until solution becomes clear. Let the flask cool for some extent. Now add 7 to 8 microliter ethidium bromide into the gel flask. It must be noted that ethidium bromide should be handled carefully because it is carcinogenic dye. After adding the ethidium bromide, now assemble the gel caster to pour the gel. Place comb in it to make wells. Now pour the gel in the caster carefully. When gel will be solidified, comb will be removed and gel will be placed in the tank. Add 1 xTAE buffer into the gel. It should be noted that same buffer will be added into the gel tank that was used for the gel preparation. Add the buffer to the level that gel should be completely dissolved inside the buffer. For gel loading, 2 to 3 microliter of 6x loading dye and 4 to 5 microliter of DNA template will be taken in PCR microfuge tubes. Mix the DNA with loading dye with the help of pipette and load the sample in the gel. With the help of micropipette, mix the contents of loading dye and DNA template and then load into the gel. Similarly, all the DNA samples will be loaded in the gel. In first well, you will load the DNA ladder in order to quantify the size of DNA. After gel loading, cover the tank, plug in the electrodes and turn on the power supply. Gel will be run at 90 volts for 30 to 35 minutes. When gel run will be completed, turn off the power supply and take the gel out of tank.
Now we will visualize the gel in the gel dog.
We will observe the gel UV light in the gel dog. Here we can confirm the DNA through the bands on the gel.
quality assurance and quality control are critical while working in the molecular biology laboratory. And before proceeding to the downstream applications like PCR amplification, molecular cloning, sequencing, and other techniques, we need to measure the concentration of extracted DNA. So in this practical, we will demonstrate that how to measure the concentration of DNA in a sample using nanodrop UV visible spectrophotometer. Basically, nanodrop works on the principle of spectrophotometer, which is that absorbance is directly proportional to the concentration and path length. So to quantify the DNA in sample, we need pipettes, pipette tips, DNA sample, blank sample, which is nuclease-free water. The purpose of nuclease-free water is to blank the instrument. And why we are using the nuclease-free water? Because we have stored the DNA in nuclease-free water. So we will blank the instrument with this nuclease-free water. In the end, we have nanodrop UV visible spectrophotometer. When we will on the instrument, it will take some time to initialize the software. When the program will set up, it will show you different samples which can be analyzed, like double-stranded DNA, single-stranded DNA, and RNA, and even protein samples. So basically, this nanodrop has the LED screen pre-installed in it, and this is the paddle of the nanodrop in which we will pour the sample into the lid. It should be remembered that while performing the experiment, you should clean the tip before pouring the sample. After pouring the sample, you will just close the paddle and run the measurement. Then it will give you the value of concentration of DNA. The system which is installed in this nanodrop is Accelero software, which is much sensitive that it can give the concentration of the DNA at the nanodrop level. It also tells us the amount of impurities that are present in our DNA sample. While performing the experiment, first of all, we have to turn on the nanodrop instrument. When the program setup will be initialized, we have to then select the type of nucleic acid molecule for which we are going to measure the concentration. As our sample is double-stranded DNA, so we will select the double-stranded DNA option on the nanodrop. When we will click on the double-stranded DNA, it will show us to load the blank sample. So what we will do, we will open the paddle of nanodrop and clean the both tips carefully. After cleaning the tips, we will load the blank sample, which is 1 microliter nuclease-free water. This is the nuclease-free water, and we will use it to blank the instrument. After pouring the 1 microliter of blank sample onto the tip, we will close the paddle of the nano drop and click the blank. It will start blanking. After blanking, nano drop will show us to load the sample. We will then load 1 microliter of DNA sample and then measure the concentration of the DNA. After pouring the sample, we will carefully close the paddle and click on the measure. It will start measuring the concentration of the DNA in the sample. Here you can see the result. There are 3 ratios. One is nanogram per microliter, which is the concentration of the DNA. Another is the A260 by 280, which gives the purity of the DNA, and again A260 by 230, which also tells us about the contamination present in the sample. So in this sample, we can see that the concentration of DNA is just 37.4 nanogram per microliter, and the purity of the DNA is about 1.53. Normally, the purity of the DNA sample is at 1.8 ratio. This graph also tells us the purity of the DNA. It should be remembered that nucleic acids give maximum absorbance at 260 nanometer. So there will be a peak on near 260 nanometer, which will show the presence and then tell us the concentration of DNA. Likewise, we can also run the second sample to check the DNA concentration. Again, we will clean the tips and add 1 microliter of DNA sample. After pouring the sample, we will close the paddle and click on the measurement. The nano drop instrument will measure the concentration of DNA. And here you can see that in sample 2, the concentration of DNA is 31 nanogram per microliter, and the purity of the DNA is again 1.49, which is A260 by 280 ratio, and these are the other ratios which shows the presence of contamination in the sample. Normally there can be 3 types of contaminants in the DNA: phenol, guanidine and protein. If the value of A260 by 280 ratio is 1.8, then it shows that the sample contains the pure DNA. But you can see that here in these results, there is contamination of phenol or proteins, which just shows that how we can measure or access the contamination through the nanodrop spectrophotometer. So that's how we run multiple samples to measure the quality of DNA. Now I will discuss the second case, that if there is no DNA present in the sample, then what values will be visualized on the nanodrop instrument. We will again clean the tips with fresh wipe and then load 1 microliter of DNA sample. After pouring the sample onto the tip, we will carefully close the paddle and start measuring. So in this case, you can see the values on the nanodrop instrument. We can clearly see the difference between the peaks of these 3 samples. Like in first sample, you can see there is a sharp peak at 260 nanometer. Likewise, in the second sample, there is again the sharp peak nearly at 260 nanometer. While in the third sample, there is no peak present, which clearly confirms that in third sample DNA is not present. So we will not proceed with the third sample for our downstream application. So that's how we assure the quality of DNA through nanodrop UV visible spectrophotometer. If the DNA concentration is satisfactory, then we can proceed for our downstream applications like PCR amplification and sequencing. One particular precaution about the nanodrop spectrophotometer is that we have to be very careful in cleaning the tips of that instrument, because it is much sensitive to detect the concentration of DNA in each sample.
In today's experiment, we will learn how to synthesize cDNA using RNA as template in molecular biology lab. After the RNA extraction, our next step is to synthesize cDNA, because cDNA serve as a template in variety of downstream applications in RNA studies such as gene expression analysis. So synthesis of cDNA is usually the first step for various protocols in the molecular biology lab. We can synthesize cDNA in the lab by using a simple PCR technique we call as reverse transcriptase PCR, or simply RT-PCR, using an enzyme reverse transcriptase that is basically an RNA-dependent DNA polymerase enzyme isolated from the retroviruses. So let's see that how to perform this experiment stepwise in the lab. For an ideal cDNA synthesis, we need RNase-free environment. So the practices that need to follow to prevent RNA degradation and for optimal cDNA synthesis, we need to wear the powder-free gloves, to use the nucleus-free reagents and labware, and decontamination of your working area.
For this experiment, commercially available reagents and enzymes are used. These reagents are extracted RNA sample, random primer, nucleus-free water, DNTP mix, reaction buffer, ribolock RNase inhibitor, and revert 8 reverse transcriptase. This experiment is performed in 2 steps. For the first step, place the PCR tubes into the cooling rack. Label the PCR tubes accordingly. Set the micropipette to 10 microliter. Now open the lids of the PCR tubes. Take 10 microliter of RNA sample and then transfer it into the PCR tubes. Discard the used tip into the waste box. Now set the micropipette to 1 microliter. Take 1 microliter of random primer and transfer it into the PCR tubes. Discard the used tip in the waste box. Add 1 microliter of nucleus-free water into the PCR tubes. Close the lids of the PCR tubes. Place the tubes in the centrifuge and spin the tubes for a few seconds so that any reagent attached to the wall settle down in the bottom of the tube. After centrifugation, put the tubes on the cooling rack. Next step is incubation at 65 degrees centigrade for 5 minutes. Switch on the thermal cycler and set the required conditions in it. Place the PCR tubes in the thermal cycler for the first step and close the lid of the thermal cycler. After setting all the required conditions, start the incubation. Take out the PCR tubes and immediately place the tubes on the cooling rack. For the second step, now add the remaining reagents in the same PCR tubes. Set the micropipette to 4 microliter. Take 4 microliter of reaction buffer and add it in the PCR tubes. Set the micropipette to 2 microliter. Take 2 microliter of DNTP mix and add it in the PCR tubes. Discard the used tip in the waste box. Now set the micropipette to 1 microliter. Take 1 microliter of ribolock RNase inhibitor and add it in the PCR tubes. RNase inhibitor is used to protect RNA from the activity of RNases. Discard the used tip in the waste box. Take 1 microliter of Revert 8 reverse transcriptase and add it in the PCR tube. Reverse transcriptase, also known as RNA-dependent DNA polymerase, is a DNA polymerase enzyme that synthesizes DNA using RNA as a template. Discard the used tip in the waste box. Close the lids of the PCR tubes. Spin the PCR tubes in the centrifuge for a few seconds. Place the PCR tubes into the thermal cycler. Set the PCR conditions as shown on the thermal cycler screen. Start the reaction and wait till the whole process is completed. After completion, store amplified DNA at -20 degrees centigrade or below until further use.
So dear students, this was all about cDNA synthesis using the RNA template. Now we will quantify our cDNA using the nanodrop, and then we will store it at -80 degree for the further use.